For the last two sessions of the day, we have starting off with genetic stability in cannabis, genomic innovations for consistent and resilient cultivation. Mike Catalano is going to lead this off. So Mike, take it away.
Good afternoon, everybody. Thanks for coming to our session this afternoon. Hopefully you had a great lunch and everybody is awake and not getting the after lunch snoozes here. We'd like to do our session on genetic stability in cannabis, genomic innovations for consistent and resilient cultivation. And our purpose today is really kind of explore a little bit the concept of genetic stability or stability in general in breeding, just to differentiate a little bit from traditional sort of trait selection or breeding for a particular phenotype outcome in plants. My name is Mike Catalano. I am the head of genomic services at Medicinal Genomics. My background is I'm not a scientist, I'm an engineer and have been mostly involved in the technology side of next generation sequencing. I've known the McKernans and worked with Medicinal Genomics, and before that Cortigen, doing sequencing for pediatric neurology. And prior to that, I was with 454 Life Sciences, where we developed the world's first next generation sequencing system. And I, back in 2011, had the opportunity to work with Kevin and sequencing the first cannabis genome. So we had a sequencing center at 454 and did a lot of work with some really interesting folks, groundbreaking sequencing work. And after Kevin had finished with ABI and had a little time on his hands, he went to Amsterdam and extracted some DNA in his hotel room from ChemDog and brought it back home. And we did some sequencing. So 454 was acquired by Roche at that time. And you have to imagine that the Swiss folks that ran the company were a little hesitant to bring anything that had the word cannabis into our laboratory, but we assured them that it was really just DNA. So happy to have help participate in that kind of groundbreaking work. I'll let everybody else introduce themselves, and those who go down the line here.
Sure. I'm Dr. Lasse Schulze. I'm the Senior Director of Flower Product Development and Agronomics for the Kronos Group. We are a large international operating publicly traded company, and my job is to support our growing sites from an agronomics perspective. I'm also running our breeding program and ensuring that our product portfolios are tailored to the different countries that we are operating in, since the Israeli clientele is very different than the Canadian versus Germany. So it's a very exciting work that we do. So we are covering literally everything from investigating light dynamics on cannabis, terpene profiles, all the way to tissue culture, plant breeding, molecular biology. That's all what we are covering.
Hi, I'm Alisha Holloway. I'm the Chief Scientific Officer at Phylos. We are breeding cannabis like you would many other crops and developing seed-grown F1 hybrid varieties. We also have a program that's focused on developing varieties with high levels of rare cannabinoids like THCV, CBC. I've spent my career trying to make the connection between phenotype and genotype. Before I was in biomedical sciences, so looking at diseases and trying to understand the genetic basis. And for the past 10 years, I've been working in cannabis and kind of doing the same thing, where we're trying to connect the phenotypes that we see in the cannabis plants to the genetic basis, and using that to apply tools that are used in other crops to breeding. So we're using marker-assisted and other types of selection to breed our cannabis varieties.
Hi, everybody. I'm Zachary Wise. I'm the VP of Cultivation for Happy Valley in Massachusetts. We're a vertically integrated operator in the state of Massachusetts. I oversee our breeding program and our cultivation operations.
Hi, my name is Maria Rojas. I'm the Director of Pathology at Conception Nurseries in California. I have been in plant pathology over 25 years. I work in plant viruses for at least 20 years. Many different types, but with emphasis on the single-stranded DNA viruses. I look into virus movement. I use many techniques to try to follow and understand viruses inside the plant cells with fluorochromes. And I also had a lot of experience in doing evaluations from breeding for disease resistance against different pathogens, fungi, bacteria, obviously viruses. And we established many different types of screenings at the greenhouse level, where you get very homogeneous input of the pathogen in there, so that your data is more reliable, especially if you're doing QTL analysis or something like that. I've been with Conception Nurseries, which is a tissue culture company, for the past 3 years. And in there, I have established several methods of indexing, and also trying to come up with better ways to have clean plants. And I established a meristem system that we use very, very efficiently right now. I will be talking about that tomorrow. And enjoying having collaboration with the breeders, helping them doing some of the screening too.
Thank you, everybody. Our first question, we want to start off really at the foundation here and say, can we explain what genetic stability means in the context of cannabis cultivation, and why is it so critical for consistent product quality? Want to start, Lasse or Zach? Or?
Sure, it's a loaded question and not so easy to answer, but I will try. I think there's a genetic component and also more like a phenotypical component there that leads to the end product. In cannabis cultivation, genetic stability refers to the consistency of the genetic makeup of the plant across generations that result into uniform traits. This is, I think, a very important component to it. And it relates to product quality because the consumers are asking for consistent products. I just came out of Israel about 3 weeks ago, and I talked a lot to pharmacists there and medical practitioners. And the very foremost feedback that I got is that consistency is absolutely key. It's a medicinal market over there. So for the patients, it's very important to have batch-to-batch reproducibility of the cannabinoid and terpene profiles. And this is something that is unfortunately not always there in the market. But for the medical markets that we are operating in, this is one of the key important traits that we are aiming for. But also for the recreational market, consistency is very, very important. We know it from a lot of commodity markets and adjacent industry, how important consistency actually is. We need to ensure that every product is the same every single time. This is at the foundation of any product that comes from the agriculture and horticultural industry. So I think genetic stability from that perspective can be described also as uniform performance. That underpins then also a brand's recognition, and is also often a requirement, especially in regulatory environments where the cannabinoid and terpene profile needs to be the same. There's also then, of course, like a phenotypical component. And I think that what is often neglected is that the phenotype is the genotype plus the environment. So the environment is heavily impacting the expression of the genotype. We have sent genetics all around the world, grew it there, and the same genetic makeup was behaving very differently in the respective growing environments. That is important because it relates also to a breeding goal, which I call robustness. It's often neglected breeding goal, but it's very important to look that the phenotypical plasticity of your genetics is not that high. So it's important to grow also in different environments to ensure that the traits are or remain mostly the same. What we noticed with genetics that we have grown in different environments is that the qualitative traits like the THC levels are mostly impacted by phenotypic plasticity, whereas more qualitative traits like the ratios between THC and CBD or the terpene profiles, they remain mostly unchanged. That's a very important consideration. So when we are talking about genetic stability, we need to look at it from a genetics perspective, but then also from its phenotypical expression.
Is this on? Yeah. So to just piggyback on what Lasse said, for a vertical operator, and as we try to expand our brand, we really need the varieties that we're working with to express and deliver very similarly across multiple different cultivation locations. And adult use markets are crowded and very competitive. And what we see is customers will quickly turn to another option if they can't get consistency from batch to batch and from location to location. So as we grow our company and we grow and expand, we're heavily focused on genetic stability from a phenotypical kind of angle where the customer experience is going to be the same no matter where they get our product. And it's become the main challenge and the focus of our breeding program.
Anybody else want to add it to that? I can say, how do we talk about the stability of the product, and we mentioned it as either genetic or characteristics, robustness. How can we quantify that, or at least qualify that, so that we have a way to measure it going forward when we're doing the breeding?
Well, I think Lasse touched on it a little bit in terms of just like the ratios of cannabinoid contents and terpene profiles being similarly expressed from different cultivation locations. And like one of those challenges is that if you take something and grow it in a greenhouse, or if you grow it in a field, or you grow it indoors, you may see very different cannabinoid profiles start to emerge from the different environments. And we have noticed that with certain things that we're working with, we tend to just see a more solid, stable expression across the different environments, and that is something that we're slowly selecting towards.
From a genetic perspective, we also can measure stability using genomic tools like DNA marker analysis, looking at allele frequencies across the chromosomes to see what the degree of homozygosity actually is. A high homozygosity rating would indicate a more stable genetic and also hopefully a more stable expression of traits, versus more heterozygous genetic will segregate in the next generation. This is something that we can measure with molecular tools. Phenotypically, this unfortunately does not exist in the cannabis industry yet, but the regulators of adjacent industries use DUS criteria. It is distinctiveness, uniformity within a population, and then stability across generations. This is a very common criteria in other crops, and I hope that it will become the reality also in the cannabis industry, because these criteria are also critical to describe a genetic or a variety in a way that is being recognized then also worldwide, that, okay, the wedding cake is going to be the same every single time. This genetic is robust enough, independent whether it's grown in North America or in South Africa. The wedding cake is a wedding cake, independent where I'm going to grow it. If you buy a Granny Smith apple, you can make sure that it is going to be green, it's going to be tart. It's a very well described, it's a system that works really well in other agricultural crops, and I hope that this will lead to better stability within the cannabis space, and it would be certainly good for the end consumer and medical patients to have a reliable product every single time.
Thank you. What are the biggest challenges we face trying to achieve genetic stability, particularly when you have issues like inbreeding depression or hybrid vigor or genetic bottlenecks? I don't know, Alisha, you want to take that one?
Yeah, I can start off. When I think about what a cultivator would hope for, they would like to see that the plants are consistent and uniform in the expression of cannabinoids and terpenes, but also in the morphology of the flowers, the structure of the plants, so that they can spend time optimizing for more yield or something like that. So when we're talking about stability, that I'm really thinking about consistency and uniformity, not something like are the genes, is there some kind of meiotic issue or something at that level. And so cannabis is an outcrossed species, and so there is a lot of concern about inbreeding depression. And what I would say to that is that it's really a numbers game. So if you have 20 different places in the genome that you have to be concerned about having deleterious alleles, then you just have to grow more plants to find those that are not experiencing issues with inbreeding depression. And so what we've been able to do in our breeding work is to make sure that those numbers are high enough that we can inbreed plants to a sufficient number of generations to have the parent lines that you would need to create F1 hybrids. And so while I do think that inbreeding depression is something that needs to be considered, it doesn't appear in our efforts and in others' efforts that it is a limiting factor for breeding cannabis. I was really excited to see one of our evaluations, and we have a mixture of different stages of the breeding process in every evaluation. Some of them are the first developmental crosses, the pheno hunts, and then some of them are parent lines at various stages of inbreeding. And I'm looking at these plants and I'm like, this one is beautiful. This one must be an F1 hybrid or a developmental cross. And they're like, no, this is actually a parent line that's inbred and it's got just a few percent heterozygosity. So it is possible to escape problems with inbreeding depression, but definitely something to consider.
You want to add, Lasse?
Yes, certainly. Like the more you are screening, you can lower the effects of deleterious genes that accumulate in inbreeding. That's certainly true. Another limiting factor that I'm seeing is there are studies that do show that the gene pool that breeders are using in their programs that drive THC up is much, much smaller than, for example, in fiber-type cannabis. That is a little bit what worries me, that are we actively, as a community, narrowing down the gene pool that we are using in our breeding programs just to drive THC higher and higher and higher, knowing that this is the biggest sales velocity driver that we know. So there's a chance that especially with diseases, and I think Maria will touch on that, with the diseases, we might run into some genetic bottlenecks where we are just seeing not enough allelic variation to breed for resistance in the high potency genetics that we are working with today. So that is one of the challenges I think that we need to overcome, but also a fear that I have that we are, as a community, just focusing on high THC, narrowing the gene pool that we are working with.
So can we go too far, and then what are those trade-offs that we look at when trying to balance hybrid vigor versus trait consistency or stability?
Can you ask that one more time?
I say, what are those trade-offs that we are making when we try to balance hybrid vigor versus trait consistency?
I guess I would kind of flip that around and say that in the breeding program, you have to take a holistic view of your germplasm bank and make sure that you have a lot of diversity, and not necessarily within particular plants, but throughout your germplasm collection, that you have representatives from widely different populations, and just to keep an eye on that so that you don't narrow the pool too much. So it's just, you know, kind of trying to have a broader view and really directed, careful planning that allows you to maintain that diversity.
Is there a plant pathology perspective on that?
Sure. So from the pathology point of view, I'm pretty biased. I'm a pathologist, I'm not a breeder, but I work with them. So one of the biggest things that I notice is you have to establish a consistent trait measurement. By that, I mean, once you have determined with whatever loci genes that you're trying to do, let's say for you're breeding for a specific disease, then you have made the bridge and the link between your loci and the phenotypic expression that you're hoping to reach. And you have all those symptoms figured out under control. Then you can use genomic tools to do a pre-screening. And so in a high throughput system where we are challenged to really push for cultivars, they require high cannabinoids, high terpenes, but also resistant to a specific or several pathogens. Then sometimes we forget the pathogens are a biological organisms, and they adapt very quickly to different pressure situations. And so if we have a control environment where we do all those set of screenings, we might get an unpleasant surprise when we switch, and I think you touched a little bit on that. Do this in different scenarios where you have your material exposed to different pressure of natural pathogen, or coming in with a very ready and stable screening method. They leave you no doubt. You measure the amount of input that you're putting in there, and you have a complete list described symptoms like an SOP, where you know exactly what to expect. So in that case, doesn't matter how you have been managing your gene pool, trying to not leave outside of your range the genes that you're working with, you're gonna have a more reliable base to select early for the undesirable traits. Whatever is susceptible, get rid of it. And then you make your pool stronger, and then you move from there with the genetic tools. But then don't forget to come back and test again. And test again in different environments at different pressures. And you're gonna have a better result doing that. Sometimes under the pressure that we are are under now, we forget the biology. And pathogens are so smart. They adjust and they evolve to say, oh, you're putting me just these resistant lines with this. So guess what I'm going to do? I'm going to mutate and I'm going to overcome you in some way, how? And I'm going to enter this way and I'm going to linking some type of symptoms that I'm going to disguise you. Or somebody who was dormant all of a sudden decided, oh, what happened here? It's not the same house for me. It's not the same type of plant for me. I need to create something different. And they're going to respond. And we have to keep that, it's traditional, it's old school, but nothing beats biology on that. We have to keep that in mind.
Thank you. Can we talk a little bit about the tools that are available and how we can use them to kind of accelerate our breeding process compared to more traditional methods, which are very widespread in use in the cannabis industry now?
Yeah, I think this ties in really well to what we were talking about in the last question. So we use marker-assisted selection for in our breeding program, and so we have a variety of tools that are common in agriculture. We have high throughput, large number of genetic variants that we look at for discovery, and then we kind of narrow that down to a few markers that we can use in breeding. And it's the value of that is that we can avoid things like inbreeding depression or linked deleterious traits, so we can select for the things that we want, but we have the information about the surrounding genomic landscape so that we can avoid issues with linkage drag. Let's see, the other thing that I was going to talk about is just the power of using those tools is so important because in a breeding program, everybody here knows how much time, space, and energy it takes to cultivate cannabis plants, and we can use these markers at really early stages to select the plants that will have the traits that we want, so we're not growing the rest of them to maturity. And then it also gives us an advantage in the probability of selecting for not only the traits that we have markers for, but having enough plants left to evaluate that we can select for the traits that we don't have markers for that are harder to get at, things like yield, or even potentially disease resistance. If you look at the probabilities, say you even want to stack two traits together, and say it has to be homozygous for the trait. So only 25% of the plants are going to have the state that you want, the genotype that you want at one locus. Say you stack another one on top of that, you have to multiply 25% times 25%. You're already at 6% of the plants. Only 6% are going to have the two traits that you want. And that's not even thinking about all the stuff else that you want in the plants. Maybe you want a diversity of terpenes, and you want to have purple flowers, and you want to have high yielding. And so instead of just looking at a few plants and only say you start with 100, you're down to 6 with the markers. So say you can look at 1,000 or 10,000 and select for the two markers, you end up with 100 or 1,000 plants that you can then evaluate, and for that whole other suite of traits that are important to you. So these tools are common in other agriculture, but I think that they're really powerful for us in cannabis. We know how difficult it is to get funding for R&D and to develop these lines. It's a long road to develop new cultivars, and so I just think that they're what's going to allow us to make the advances we need to make to develop new varieties.
So I just like to add to the comments that we have made before about the gene pool and pathology. So you stated very nicely about the markers. And if we don't as you are doing your generations and generations and selection, and you might leave some very important loci that only work together under normal conditions that might not be expressed when you are testing them for a particular pathogen, for example. And then you lose some of those for your final product. And those are one of the unexpected surprises I'm talking about. You know, all that super hard work that you have done, and then at the end, your material isn't that good as you expected it. So one of the points that I want to bring today to this panel is to not forget that the triangle they always learn when we go to school about plant pathology, that is comprised of the pathogen, the environment, and the genetic, the host genetic stability. And if we only focus in that, we're going to be losing some of the gene pool. And by doing that, then we're going to be losing some of the potential resistant genes that we might have acquired at the beginning, and we didn't know we lost, because maybe there's some type of relationships that are not phenotypically expressed that we're undergoing, and we weren't able to screen for and keep. So I think markers, if we increase the number of markers that we can get with different phenotypes, are a tremendous tool to help on that. Yeah.
Else want to add anything? Are there unique advantages to focusing on genetic stability for seed production over clone production?
Yeah, so we're focused on our breeding program and making seeds because of their ability to store, ship, and scale, cost effective. Where we're not making any claims that we have achieved our goals, because we have very lofty expectations for our seed stock, and our customers have very lofty expectations for the quality and reproducibility of what they're getting across markets. But we certainly love the fresh start that seeds give you. I mean, you can go to tissue culture, and you can go to meristem culture and try to reset your genetics and remove pathogens that way. But I think it becomes easier and more cost effective if you can find the right seed stock to start from. And it certainly is a scalable solution for cultivation and for cultivators. But it's a challenge, and we've spent years working on lines that have gone to dead ends for us, and we've had successes in other areas as well. But we're utilizing marker-assisted selection for some of this stuff. But as new tools come online, it looks like the pace and speed at which we'll be able to achieve these goals is going to change rapidly, and I think we're starting to see that. But for us, we still do a lot of clonal propagation. It helps us give our customers what they want at the retail level, and we're not against resetting our mother stock from meristem tissue culture. But when we can plant a whole crop from seed and watch how it grows with such vigor and such strength, and yeah, it's very impressive. So I think seeds are the future.
I have to defend the clonal because I'm coming from a tissue culture. I'm just going to give you my perspective. But honestly, I think there is room for both of them. There is a need for both of them. And we cannot just thinking of moving on to seeds, because clonal is strategic and clever right now, and it's going to continue for a while. If you start with you start with clones, and I'm talking about what we produce, like zero clones, like generation zero, 100% pathogen-free, pest-free, highest quality you can get, and 6 to 8 inch little plant. And if you put that plant under conditions, for the purpose of this example, let's think indoors, because for outdoors it's a different story. So let's think of indoors, and then what this type of material is going to give you is very consistent genetic stability, very uniform morphology. You're going to have very uniform results with the flowering, the time to flowering, and not even mentioning the terpenes and the THC. Everything is going to be, they're clones. They're going to behave that way. And they're easy to handle. So then from the operational standpoint of view, you already have a head start because you don't have to germinate them. The plants are already there, ready to go. So you win time there. And so your time to have these babies into market is going to be shorter. And so what that allows you is to have more generations in a year for that if you were to start from seed. And then apart from that, then you're going to reduce your cost because it's less labor, and you're going to increase your profit. Sorry, the English suffers there. So I think that way. That's why clones. I'm not talking about clones starting from a mother plant. I'm talking about generation zero clones, right? With that, starting from a mother plant, that's a completely different story. I think that give you a tremendous start because you start from extremely clean point of view. Mm-hmm.
Yeah, I think that you make really good points. I have to defend seeds a little bit, but I agree with everything that you said, and I think it's really important to think about the difference between a mother room and what you're talking about, Maria. They're really different beasts. On this, and we have part of our breeding program is in rare cannabinoids, and that's all clonal based. So the seed breeding that we do right now is just for the THC varieties. And so I agree. There's place for both of them depending on your goals. But I do have to defend seeds a little bit. So what we've seen is that when we look at seed grown versus cloned, we're able to match the cycles almost at the same time, and we see that we're having to suggest to customers that they plant their seeds about a week later than if they were going to cut clones. Now that's a mother room, not a tissue culture clone, because the seed grown are so vigorous that they need a little bit less time in veg. So I think cycle time can be roughly the same. The other thing that has been really exciting, I'm a researcher, that's what's in my blood. But what's been really exciting on the breeding side in discovery is that when we have a new marker, we can very easily go into our inbred parent lines and introgress those traits into the parent lines. And that's really hard to do if you're only working with genetically variable clones. So we have this power to do marker-assisted backcrossing and bring in new traits as we find them. If we find disease resistance, we can bring that in. If we, you can name your trait, but there is a lot of power in having those inbred parent lines and the tools that you can apply.
I think that both have their validity. Tissue culture certainly has in the last years gotten much more traction, and we are now at the brink of having some stable seed lines commercially available. Both have their validity. What I believe is transpiring in the cannabis industry is that the stable seed lines or genetics coming from stable seed lines will be these genetics that cater to the mass market. It's going to be the Coors Light. These are going to be very, very established genetics. The genetics that come from clonal production, from tissue culture, are more like the craft brew type, what's the next best thing genetics. And this is just due to the nature of developing stable seed lines and genetics that come from that are stable. It takes a long, long time. So you have much less flexibility to come up with something new, something new that you want to discover. This is what I believe is going to happen. And also, quite frankly, not everybody has the ability to run a stable seed line program. People don't have the funds. We are going to touch also on the actual cost involved in working with genetic markers, and it's not available to everyone. But I believe there will be also in the future some solutions to that. So I believe both have their validity. They are not going to go away, but they serve their own purpose. If we could move everything to seeds, I think there's not a single cannabis cultivator that would say, well, I would like to stay with clones. Seeds are easier, they have a higher degree of automation, but both will have their validity in the future.
Thank you. Our final topic is based on, cannabis industry as a whole has been a bit slow to adopt, genomic assisted techniques widely compared to traditional agriculture. Kind of uses this in a pretty regular fashion. How can we overcome these practical challenges? The two biggest ones being cost and access to expertise, to make these tools a little bit more widely available for breeding in our industry. Open to the group.
Well, I can start. Sure. I'm currently operating in a market where we're seeing a lot of price compression and market saturation, and it makes it very difficult for us to allocate funds to research and development. But we also see it as an essential path forward for our survival in the space. So we do what we can, and of course, Medicinal Genomics as a partner in that has been excellent and very accommodating, working with us, even though we maybe aren't sequencing as much as Phylos is. We're finding a way to squeeze it into our budget because we know it's essential to our survival. But in terms of answering the actual question on how to overcome the practical challenges, I'm not sure that I have a solution. We need markets to stabilize and we need price compression to stop. I mean, over the last almost 20 years of cultivating, the cost of producing a gram of flower has just risen for me, and the actual value of that flower has just gone down year after year after year. And the consumer has come to expect more for less. And I don't have a solution to that problem.
I think one of the big challenges also to adopt genomic tools is also cultural. A lot of plant breeders from the prohibition era are considering plant breeding or cannabis breeding rather an art than natural science. And there's that the notion of, well, science and genomics is something that I rather don't want to touch because it goes against my intuition to use these tools. Like I'm like an artist in my area of cannabis breeding. And I believe partnerships with breeders that come from this era that have this attitude will, I think, break down these barriers more and more, making them understand that this is an actual tool that is being used also in other industries for a very, very long time. I also believe that the notion of genomics being something evil, there's a misconception that and misunderstanding of what genomics actually means. most people, when they think about genomics, they're thinking about GMO, evil, I'm gonna grow wings when I'm gonna consume it. This is just not going to happen, and you need to understand and advocate more and spread the understanding that genomics entails a wide array of different techniques and tools that are available. I think a lot of people also don't understand that when you're doing a simple gene edit or a silencing of a gene, that this is actually a much more precise and less invasive way to change the genome than normal breeding, where you're creating way more off-target mutations. When you are growing a plant, when you do breeding, you're dealing constantly with any type of mutations. So genomics is quite often much less or creates way less alterations within the genomic makeup of the plant than traditional breeding would. But that knowledge is just not there in the general public, and paired that with a cultural hesitancy, thinking that's more an art than a science, I think bridging that gap paired with some educational component will go a long way for genomic tools being adopted.
Great, thanks. Anybody else want to add anything?
I just had one other little thing. Just kind of, I was taking this little trip down history lane for a second, and I've only been in this industry for 10 years, and I know, many people in this room have been in the industry for longer. But when I first entered the industry, there were a lot of giggles when I talked to people about what I do, and now that does not happen anymore. There were times when I tried to partner with universities where they could not say the word cannabis for fear of having their federal funding pulled down. I think that though, you've got Mike in the audience, you've got Todd, you've got others in the audience that are at public institutions, and you're doing research that is moving this industry forward. And so public-private partnerships and or just university research is going to drive this field forward.
I just want to add an idea with, and this is about the genomics, is how do you talk about genomics to a grower? Or how do you talk about the customers that are trying to protect what they have, and they want to be up to speed with what everybody talks about, genomics, name it, you got it. So one idea that crossed my mind is to try to tell them, just select one clear item that you want to work on. Let's say disease resistant, obviously, mine, but flower structure or the cannabinoid profile. Just choose one and start working with something that is not that expensive at all, and try to get some SNPs done, some sort of knowledge, some genotyping by sequencing, and then try to work with the people that do this all the time, consult with them, and then you have an idea of what your population is all about. What do you have? And then from there, you can seek a strategy and protect your thing. So it's all about education. Don't get that feeling that if you don't rush into this, you're out of fashion. I think it's the opposite. It's just education, but go one step at a time, and we're all gonna get there.
Great. Thank you, everybody. We have a few minutes for questions, so if anybody has something they want to ask, please raise your hand and we'll get you a microphone.
Thanks for having me. I appreciate all the information you guys are providing. I'm going to ask this question as respectfully as I can. I'm trying to speak for the independent cannabis citizen scientist community. My question is for the representative from Phylos. There's a dissonance in the community I just previously described regarding Phylos. I was wondering if you could clarify what led to the dissolution of the nonprofit organization formerly known as Open Cannabis Project. And secondly, could you explain your company's previous gene acquisition policies and strategies, especially pertaining to the terms and conditions for acquiring the genetic source materials being used in your for-profit or just research R&D projects?
Yeah, I wasn't involved in the Open Cannabis Project, but I know that they didn't quite have the follow-through that was hoped for. No, it wasn't a subsidiary. There were some connections from some of the staff. So we've since the beginning, I came from an academic background, and I really believe in sharing data. We've shared thousands of data sets from genotyped plants, and those have been used in publications and in other. Yeah, so we. It is. So we. There are two separate things. So we did a lot of genotyping in the early days, and those were put into what was called the Galaxy, and those are the data sets that we've released to the public. When we got approval from whoever gave us the sample, they would allow us to release the data publicly. Some folks decided not to have the data released publicly, so we weren't allowed to put those forward. And so that's one set of data where we got small stem samples that we genotyped and then put them into the Galaxy. And then we also have a germplasm collection that is part of our breeding program that we started in 2019. And all of the in-licensed germplasm comes with an agreement from the person that either sold or licensed us the material. And so that comes with the ability to breed with the material, and that's how we've extended our germplasm collection. We're one of the only companies that I know of that's licensed and paid breeders back for the work that they did before we got the material. And I think that you'll find that our licensing agreements are common in agriculture, the kinds of work that we do. Oh no, that's a separate thing. So when we did the initial genotyping for the Galaxy, that was receiving small pieces of dried stem, because at the time that was what was legal. You had to wash the stem, get all the cannabinoids off. It was from a dead piece of plant material, and then those were genotyped and used to create the Galaxy. But what's in our germplasm collection was in-licensed with agreements from breeders. So those are two very separate things.
Excuse me, may I ask a question now? Maria, my question is for you. So you're a virologist working on cannabis. As you know, there's a long list of different viruses reported on cannabis. They're found. I'm not talking about viroids, HLVD. I'm talking about specifically about viruses. Cannabis cryptic virus, we have alfalfa, we have tobacco mosaic virus, cucumber mosaic virus, leptospirosis, beet curly top. But not all of them apparently causing diseases. They are found, but we don't know if they're actually a causal agent of any problem. If you want to choose one of these viruses that is the most important one, which is worth working on the genetic resistant part of the story, what would be your choice? Thank you.
Well, okay. I'm going to start by telling you that one of the things that we do when we do intakes in our company is do a complete profile for any virus that somebody thought has seen on cannabis. And I've done this on a regular basis. I still haven't seen a sick plant from many of them at all. This is just scaredness. So many of those are not even economically important, and even for outside, where you have the vector at the aphids. Aphids would be a very efficient vector for alfalfa mosaic virus that give you a beautiful modeling. So you have beautiful modeling. It's not really going to reduce your yield at the end, okay? If you happen to have it. But if I were to choose one, just for interest, a personal interest, I would go with B. culita. And the reason for that, well, I'm biased. I mean, I'm a Gemini virologist. That's a Gemini virus. And it's transmitted by leafhoppers. But because at the end of the day, a good plant infected with B. culita is going to be stunted, the leaves are going to crumple, and that's going to hurt your plant. So if I were to choose one, I'm going to take B. culita. But how many times have you seen reported cases of B. culita, of alfalfa mosaic virus, or even TMB, and indoors? I mean, like 0 in my case, honestly. So could be perhaps because of the vectors, a problem a little bit in the outside, you know, when you go outside. In California, honestly, I don't consider them. We do the testing for them because I want the customer to feel okay with it, and because we want to make sure that we don't bring anything that is going to be carrying anything into the tissue culture. You'd never know. Unexpected surprises, we want to avoid them. But yeah, I don't think viruses are a big threat to cannabis right now.
Thank you. I think we have time for one more question, if there is one.
Yeah, I've just got one more. Thank you, panelists. I'm a customer of both Phylos and Conception, so I can tell you the growth rate between the 0 point or whatever, however you mentioned it, is almost equal. I found myself, so great vigor. My question was more about, you had talked about the plasticity in the genome and how that manifests in the flowering stage. For those of us that are still on the clone life, how can that, and I don't know if this is a dirty word, I hadn't heard it in this panel, the epigenetic changes that might occur, potentially using that word incorrectly, but with your mother stock, you know, those kind of environmental pressures and things that any of us that run large facilities inevitably encounter, have you found that indeed can create some changes in the gene line that, you know, again, manifest out in the flowering stage? Thank you. Anyone?
Is that for me?
Really, for any of you.
Okay. Okay, when we haven't really talked about mother stocks here, and the other type of clones that everybody loves to do is just cuttings, you know, and from mother stock. I guess that goes back to how's the health of your mother stock, you know, phenotypically and endophytically. And I'm going to talk about that tomorrow. I'm going to show you how does it look like. So basically, your mothers are going to be as good as your environment is. If you start with a clean start, you know, and keep that plant as a mother stock, at least for the first round, you can make all the cuts that you want. You can generate a very decent pathology-wise population. But if you don't take care of it, you have no choice but come back to gene zero clones, generate, and start again, you know, recycle your mother plant so that you can continue your proliferation of the cuts. Would that?
Yeah, I think there's a lot of, sometimes, you know, some of my colleagues will believe that there's some sort of shift in the genome, but to your point, I think it's the mom plant has been cut
You, you know, my partners can talk a lot about this too, but are you talking about somatic mutations that all of a sudden your plant looks a little bit different, you know, phenotypically? And so the question is, what happened with the number of cuts that you have? Did something get introduced there, a sort of a mutation? First of all, there has to be a direct correlation with something phenotypic and not environmental induced, stress induced, lighting induced. All of those you have to put on the side and to consider your mother as a complete entire stock. But any other comments on that? Any takes?
So we had an experience about 6 years ago where we had a Jack Herrera genetic, and measured always at about 20% THC quite consistently. And all of a sudden, it dropped to about like 12, 13%. We know that we had the tremendous amount of powdery mildew pressure at that time with these mother plants that we have not experienced before. So I believe that something happened probably with the methylation patterns that caused that drastic drop in potency. And still today, we cleaned it up through tissue culture. We made the HLVD testing, all kinds of pathogen testing with that genetic. Everything came out negative. So something has happened that caused that potency drop from one batch to the other to drop, and we were not able to get it back. So is it possible scientifically that methylation changes of the DNA cause significant impact on traits that we are interested in? I'm very convinced that that is possible, but it needs to be paired with extreme environmental pressures usually. We know it also from animal studies that methylation patterns are usually changing under extreme conditions. So if you have experienced something similar, it is certainly possible that this is the case, but we have not gone through like an actual genetic analysis to confirm that.
The only little point I'll add to that is that if you look at cannabis compared to other agricultural crops where we have a long way to go, we haven't really studied the fundamentals of this plant and its genome to the extent that we have that information for other crops. And that's really important work, and something that I hope will continue to improve with the public-private partnerships and work in academic institutions around the world. But that's a great example of the type of research that needs to happen in terms of looking at methylation patterns and maybe telomere length or other genomic characteristics that might normally get done for other crops in a lot of different places before we launch off into more complex efforts in terms of breeding. But I just wanted to thank everybody on the panel and thank all of you for attending today. We're around, so if anybody has any other questions after this, you can grab us.
Thanks, Mike.
All right, we have a quick 10-minute break, and we're back here for the last session at 4 o'clock.